for this field, the way the hydrogen atom is for atomic physics, or the way C. elegans is for nematodes, and so forth. There are actually thousands of different species of algae, and of course there are innumerable bacterial species that can swim using flagella. In fact, the flagellum is an amazing structure that is essentially the same in no matter what organism it appears. It's one of those things that was such a great invention that it apparently evolved and was incorporated into many different species. So this work was done mostly by Jeff Guasto and Hussein Kurtuldu, two postdocs who work with me in my lab at Haverford. But also, I am fortunate to have a wonderful collaborator, Carl Johnson, a biologist who is an expert on Chlamydomonas, but more on the genetic side. And that has been extraordinarily important in this work. It just was an accident to have a colleague at a small institution who was an expert on the same organism that I wanted to study. And as I will mention later, my interest in this subject began with a sabbatic leave that I took in Cambridge, England, working in the laboratory of Raymond Goldstein, and that's where I first became familiar with swimming of microorganisms. My background is in fluid dynamics, basically, especially the nonlinear dynamics of fluids, phenomena like instabilities, turbulence, pattern formation, things of that sort, convection, rotating fluids, lots of more classical fluid dynamics, but I became interested in the last few years in extending my work to the biological interface, and I must say it has been an exciting adventure, because there are some things I know that are useful and can be applied in this area, but it's also a tremendous learning experience because there's so much that's new. It's in fact so much that's new that it's somewhat humbling, but it is a lot of fun. So let me tell you a little bit about this organism. I'm going to begin my talk by giving you some information about the kinds of things that these organisms can do. And then I'm going to ask you to do a little exercise, which is to propose some investigations. So you need this information in order to do the next step, which is to design some investigations of these organisms. Okay, so first of all, they swim, but they're often found in soil. They're not necessarily found in open bodies of water, but they can be found in puddles and ponds, and they're extremely widespread in nature. It's a single-celled organism. It's a prokaryotic cell. It has two flagella. I'll show you the structure of the flagella shortly. They're about 10 microns long. They vary a little bit, but not too much. And the flagella are 10 to 12 microns long, about the same size. This is much bigger than most bacteria. If you compare to E. coli, which can also swim, that's just 1 or 2 microns in size. And has more than 2 flagella. And the flagella are different structures in these two cases. So it swims using a breaststroke with these 2 flagella like this, but it beats them at about 50 times per second. And this already is something quite remarkable if you think about it, because we're in a viscous fluid, Reynolds is low, viscosity dominates, and yet these organisms are capable of moving their flagella through the fluid at this fantastic rate of speed. In fact, the tips of the flagella move more than 1 millimeter per second. It's 100 times the size of the organism. So quite amazing the structure of the device to do that. They also have, I need to, play it for you, show you what it actually looks like. This is high-speed photography, so it's been slowed down quite a lot, but it's actually 50 times per second. These organisms are also phototactic. They can sense light. That seems reasonable since they are able to, they're photosynthetic. They do photosynthesis. Although they can get nutrients, if you put them in the right kind of bath, they can get nutrients in other ways. Yes?
So are these guys not buffeted around by Brownian motion?
Brownian motion is not very visible on the 10 micron scale. But if you put a 1 micron particle in there, you will see the Brownian motion, and I'll show that to you later. But on the 10 micron scale, Brownian motion is not very significant. It's odd that it is so short and sharp for you to find. So you don't see any objects of it. So they can detect light. They have a light spot. It is sensitive to light, and their swimming can be biased so that they can seek out places that are well illuminated, but also avoid locations that are too highly illuminated. And that whole subject of phototaxis is itself a very interesting problem and a very deep one. Okay.
I have a question. Do you know why the photosynthesis is limited to this range of visible light?
No.
Or is it not?
I mean, why? Basically, if it becomes redder, this isn't giving up much. 720 is not giving up much, because that's the red end. It's not very sensitive to the very red end of the spectrum, but most of the light intensity in the sunlight is shorter wavelengths than that. So I want to remind you that this is one of many different swimming organisms, thousands of species, maybe hundreds of thousands, I'm not sure. This is a picture of E. coli here. This is Paramecium with its cilia, large number of cilia, Chlamydomonas, and life. This is just one example, but there's a lot that can be learned. So let me give some acknowledgments here. As I said, I began this in working with Ray Goldstein's group in Cambridge. But much of what I had to say was done in the last 2 years at Haverford, and we have support from NSF for this work. But when I was in England, I had a grant from the Lever Home Trust as a visiting professor there. So this is what the flagella look like, and let's see, I have to bring my. So these are microtubules. The green are microtubules, microtubule doublets, they're called. They're pairs of microtubules seen in cross-section. And if you count them, you see there are 9 of them. 9 pairs, and also 1 pair in the center. And they form the main structure of the microtubule. It's about 100 nanometers across the microtubule. Too small to image. It's smaller than the wavelength of light. You can see them under a microscope because they're elongated, but there are it's only 100 nanometers. In between the microtubules, you have dynein molecules, which is one of the molecular motors that has been studied quite a bit. And the dynein molecules can move along these microtubule pairs. And as they move, they cause the flagellum to bend. So it's the crawling of the dynein molecules along the microtubule that causes it to bend. And it's the bending that is the source of the swimming. These are very different from the situation with E. coli, where the energy source is provided at the boundary between the body and the flagellum. Here, the energy source comes throughout the whole length of the flagellum. Okay, so that's about all I'm going to say about that. We'd like to. We hope it might be possible eventually to relate the fluid dynamics measurements to what goes on inside. Yeah.
You mentioned that the flagellas are 10 micrometers long.
Yeah.
So, and you said that the motors are along the whole flagella. So how does it deal with diffusion? I mean, is it an active process to bring the ATP, the energy, to the end of the flagella? Or?
Well, they can take in. They're surrounded by a cell membrane. The flagellum is actually surrounded by a cell membrane, and I think you get diffusion. It's not all diffusion along the membrane, but there can be diffusion transverse, directly in and out. But I don't really know too much more about that aspect. Okay, so as I said, remember, I'm giving you some information that you'll be needing, but I won't be going on too long before I ask you for some input. And what I want to show you is something remarkable, that these cells are able to spread out. And let me show you what that looks like. So here, what we've done is to take a cuvette containing some Chlamydomonas cells, and each little white spot is a Chlamydomonas cell. And let me show you what it looks like. Over time, you see that they spread out. You can centrifuge them, concentrate them, and then if you let them go, they will spread out. And one of the interesting questions is how they do that. And here's a sort of a less magnified picture of it, where they're all at one end of the cuvette, and they spread out. Well, one of the questions that we posed was whether this is a diffusive process. Is this basically a kind of diffusion, or a kind of random walk? And that's something that I'll tell you more about in a bit. I want to show you that you can track certain cells and tracers at the same time. This is a video Each this is a swimming cell, and the red are tracks left by 1 micron tracer particles. So you can actually detect and track both at the same time. And this is one way you can get information about the system. So keep that in mind. That's something that can provide data and raw material for investigations. The methods that we use for tracking are themselves kind of interesting. I won't have time to say anything really about them, but the trick, one of the tricks in tracking either cells or particles is that you need to be able to know pretty much where the particle or cell is going to be at the next time step, so you can look in that region. Because if you don't have an approximate idea of where it's going to be, you will make mistakes and identify a particular particle with a different particle in the subsequent frame of the video. So we use a predictive algorithm to be able to estimate where the next image will be of a particular particle, and that was devised by Gollub, Gale, in mechanical engineering. Okay, so just a little bit more background about flagella. So I mentioned that they're highly conserved structures, which means to a biologist that they're found in many different organisms. They're found in the respiratory system of people, structures similar to them. They're believed to be responsible for the left-right asymmetry and embryonic development in some organisms. The phenomenon of exploration or spreading out has been much more studied for prokaryotic cells than for eukaryotic cells like these. As bacteria have been studied tremendously, but these have been of interest relatively recently. Okay, so now it's time for you to make a contribution. You've seen some of what these swimmers look like. You've seen some examples of how you can get information about them. I would like you to propose an investigation that would yield insight into swimming dynamics of this organism. Okay? So what I would like you to do is to take 2 or 3 minutes and turn to a neighbor and actually discuss a potential investigation and write it down. Okay? Something you could learn that would be of interest. So, the next 2 minutes, and
And they can pass forward.
So I'd like some volunteer suggestions. I'll just make a list. We won't worry about too much about how practical they are, the difficulties, but let's just get down some ideas. Okay, who will start? You can just go around the room. Yes.
So Scott had the idea to change the viscosity and then the flow consideration. So maybe you could vary both actually. Just increase the viscosity systematically and then also put them in, for example, different types of shear and flows or something like that.
Okay. Next.
We have a corollary to that one. So if we were thinking, in addition to changing viscosity, you could change, put them in a viscoelastic environment. Basically, change the environment to see if it changes the kinematics.
Does the flow? Yes. Thank you. Yeah, so you want to know if the flow pattern, if the swimming stroke varies with viscoelastic properties, which means that the fluid can store energy. And your motivation in asking about viscosity was just what did you think might happen?
Maybe they vary their gait in a non-trivial way.
So also the stroke.
And did you say they normally live in soil?
Yes, or in damp environments. Yes, but
So
in
you
soil.
could always you could put them in like a soil material rather than a fluid.
Okay, so vary the environment, one or another. Okay, what else? Come on, more ideas. Keep going around the room. Yes.
If you put a lot of them concentrated suspensions, will they affect the mixing of the tracers?
Would they?
Affect the mixing of the tracers?
Ah. Okay, mixing. The tracers. So they stir the, how do they stir the fluid? Yes, other ideas? Keep going around. Doesn't matter. Don't be too critical. Yes?
To see if introducing a light source will change the diffusion of.
So we were thinking of actually different lambdas, different wavelengths, like.
Yeah. So, okay, there are a lot of investigations you could think of involving light. And you can vary the wavelength. You can vary a lot of things. Wavelength, intensity, so forth. Yes, please.
We're thinking of maybe a temperature gradient from one side to the other if possible. And then,
Do they respond to temperature?
yeah, then we were thinking of maybe trying to corral these dogies in a certain way where the longer ones could be some variation in length. Maybe there's a dependence of velocity on the length of the platform. So maybe find or chop off, or do something to see if there's a dependence on how fast.
Yes. Actually, that brings up a very recent paper that's just out, where someone, you can deflagellate these organisms.
I have no other way to say it.
It sounds mean, but it's not that bad because in an hour they go back, and you can study the swimming as a function of the length of the flagella as they go back. And this was just done as very interesting. I know many of you are interested in synchronization. Remember last night? Well, it turns out that if they're shorter, they don't synchronize very well. And there's a transition where they'll have to be a certain length before they'll synchronize. Other ideas?
Is it possible to make them express more flagella?
These, no, they don't. But these don't. These are only, they're strictly biflagellates. Yeah.
I'd like to see how the viscosity of the medium varies when you increase the concentration.
Okay, so you might wonder whether the organisms themselves affect the viscosity of the fluid because it's active. Because it's active. Okay, what's next?
Food gradient.
What was that?
Food gradient.
Food gradients. So, yes. Now, normally, since these are photosynthetic, they're not actually looking for food, but other species are able to sense and do engulf particles. So there are other microorganisms that do. So it's still a relevant question. Yes.
We check for non-linearities in the fluid since they're quite big for swimmers, or if you have a lot of them, if there are any non-linear effects in the fluid flow around.
You mean in the flow pattern?
Yeah.
So you're asking about the,
If there are any inertial effects, because they're quite big for swimmers, if there are any inertial effects that
Yeah.
come into play when you put a lot of them, or even when you have one, because they're quite big for swimmers.
well, they're bigger than some and smaller than others,
Yes,
of course.
sure.
But you were interested in inertia. Are there some inertial effects or not? Yes.
I'd be interested in gluing one of these
Could you
to.
guys introduce yourselves as you speak? So, I mean, just remind me of your name.
I'm CG.
Yeah.
I'd be interested in gluing one of these to a calibrated cantilever to look at the deflection of it while it swims, and that way you can try to get a measure of how much force it's exerting.
Okay, forces. Okay, so you're talking about getting really at the mechanics of how it actually works. Yes.
Could have more than one of these organisms when you have.
Yes. Yeah. If you, what do you think might? What would be the interest there, or what might happen?
They might like phase lock or.
Okay. Look at interactions. You're talking about interactions between the organisms, right? Yes.
Look at the, I'm John.
John.
And if you look at the behavior adjacent to walls, it looks like they climb the walls faster than the bulk, at least in the images you're showing. So behavior next to surfaces would be interesting to study.
I didn't know there was anything in the movie that.
No, me neither.
It's on the images.
Oh. Okay. All right. Yes, it's a question actually that we're very interested in currently, although we haven't done much yet. Yes.
If we put lots of these guys together,
Your name again?
and then
Yes. Thank you.
so if we put lots of these guys together, we can look at the statistics of say tracer diffusion. And,
Tracer diffusion in a population.
yes, and also the number statistics.
The number.
Number statistics of the guys. So
Number of fluctuation.
Number of fluctuations. Yeah. So statistics. Involving populations. Maybe I could summarize it that way. Something like that.
Converters. Okay, so what do you,
Okay, well, we could go on and on, maybe two more, and then we'll.
We were proposing to follow the kinematics of the flagella during the stroke.
Yes.
How it bends, if we can actually follow that.
Very relevant, and a little bit difficult, but very relevant, and I will show you a little bit about that. Yes, other, one or two more, yeah.
Ayushman, this is really a question. So the photo tags, for getting back to the idea of food gradient, do they have CO2 sensors? I.e., would they go towards higher concentration of CO2? That's their food.
Good question. I'm not aware that they do. As far as I know, they don't. I would have to check to be sure, but I don't know if there's been any experiment actually looking at CO2 gradients. But that's an intriguing idea. Let me put it down. One more. Yes.
I actually just have a question. Why are they swimming? Like, what are they trying to get to? They don't have natural food.
Well, and one can only speculate, right? But you can't ask them. But they may be spreading out in order to have an opportunity to explore environments with more or less light intensity. So they need to be mobile in order to do that. That's the common speculation. But that doesn't count as a suggestion so a suggested experiment, so I have to get one more, yeah.
Oh, yeah, I'm sorry, I also have a question.
I have a suggestion.
Do all algae do this? Do all algae swim?
Do all algae, no, they're not, they don't all have flagella. So it's not the, they don't all do this, yes.
A lot of bugs walk in like a level flight, so, and they keep track of the right turns and the left turns, so they don't make circles on each other. So maybe you could see if they do something.
So you're asking about the tracks,
Yeah.
and the statistics of the tracks. And I guess you'd like to know whether they, do they go in circles and not make any progress? Or do they have a more random trajectory so that they actually spread out more? These are very interesting questions. Okay, yeah.
I wonder if I introduced like a very low acoustic sound, like speakers at 50 hertz and 55, 60, that you can maybe drive them to swim synchronously, you know?
Acoustic response, okay.
Maybe, I would find it hard to believe if the only thing they know how to do is beat their wings like that, if they heard one beat. I mean, how could they avoid not beating a tempo?
Okay, that's the last one.
So I'd like to put them in a convergence in a microfluidic channel to see how they deal with traffic jam.
Yeah, so you
Or the
want to use a
geometry
higher-
of the...
you want to use a higher concentration.
Yeah, so like
And-
something converging
right.
in a microfluidic channel to see if they manage to, and how they manage to
Right,
escape.
so, for example, we were talking last night about coherent motion in bacterial suspensions. Remember that came up several times last night. You might wonder whether the same thing could happen with these guys. Will they begin to be coherent? Will they interact enough to become coherently swimming? But you had something else in mind, I could tell by your face.
I was just imagining this experience. You put them in a microfluidic channel,
Yeah.
and you make it
Yes.
converging and then diverging to see what's the.
Yeah, but what he's saying is that maybe in order to go through, they will synchronize. Or
Maybe they will
something like this.
get a corrected behavior. Yeah, I mean, I don't know what to expect.
So, look, I think, okay, one more.
Is there a difference in the bending between both flagella? Because they have kind of a chiral structure, and I would guess that both flagella are a little bit different from each other.
So you're asking about asymmetry between the motion of flagella. It need not be a completely, you're speculating that it need not be a completely symmetric pattern. And you're exactly right, that does happen, and I'm glad you brought that up, so we might just call that asymmetry. Okay, so look, we got a lot of ideas here, and it would take many years to explore all of these things. Aspects of some of these have been investigated. We've done a little bit on a few of these, and others have looked at a few of these things. But you have actually listed quite a few potential investigations that have not been carried out. Of course, you also have to think about not just whether it's sort of abstractly interesting, but what it might tell you. What it might tell you about the organism or its functioning or its energetics. So some of these, if you actually had all of these things to choose from, some of them are going to be preferable as a starting point. So when you're actually planning a research investigation, you're going to want to think of what could I learn that might have an impact on our understanding of this organism and its physics. So that's something always to keep in mind. But just a lot of curiosity-driven questions is a very good way. So thank you for those contributions. So I'm going to tell you a little bit about some of the things we've done. But I think this makes it clear that there are a lot of possibilities and many things that no one has explored yet. So, these are some of the questions that were motivating us. We saw this spreading out behavior. We wanted to know if it's really diffusion. And we wanted to know how the fact that this was low dose number affects these organisms. We wanted to know about the flows induced in the fluid and about the mixing that might be produced by that. In fact, several of you mentioned that. And we'd like to understand something about the drag forces acting on the flagella and acting on the fluid, but those are reciprocal forces. But there's also drag forces acting on the body. And all of these would be interesting to know. I mean, to have a real description of just the basic physics, you want to know something about the power generation and about the forces that are produced there. And of course, you want to understand how it makes forward progress, and we'll see something like that. So, so I'm going to present maybe 3 or 4 examples of investigations. Yeah.
Sorry, it's just a question. So if these two flagella are not the same, so then there might be a force along this axis, and the particle might actually tip over, like this, right? So does it ever tip over on its back?
It turns out that they actually swim in spirals, generally, and it's believed that spiral swimming has something to do with facilitating their phototaxis. But let me try to stick to a particular line of discussion now. So I invite you to ask clarifying questions if I'm unclear or I miss something that makes it hard to understand what I'm saying, and then sort of more expansive questions we'll save for a little bit later, so I can tell you about some of these investigations. So, okay. How do you tell if it's diffusion? And what is diffusion? So think of a random walk. You know a random walk gives you diffusion. What are some of the properties of a random walk? Well, there are, you start to think about that. When you have a random walk, one property is that the mean square displacement grows proportionately to the time. That's one property of a random walk, or a diffusive process. Another property is that it should obey the diffusion equation, which is that basically the flux of stuff that's diffusing, say the flux of the number density of organisms, should be proportional to the gradient in their concentration. The bigger the gradient, the faster the flux will be. So there are different ways you could explore this question. The way we chose was to look at this property. So I'd like to show you how we did this. And this was something that we did in Cambridge with that group of fantastic postdocs that I mentioned before. The idea of how to do it was mine, but I couldn't have done it myself. So here's what we did. Remember we had this cuvette with all the organisms concentrated, and we watched them spread out. Okay. So what we did was to measure at each distance along the direction of travel. We measured the local gradient of concentration of organisms, which we call dCdx. And we measured the flux, which is the number of organisms crossing a line. And we did this for all these different, for many different lines along here as it moves. So what happens is that this pattern spreads out and becomes wider. But at each point you can measure the concentration gradient in the flux, and you can look to see if they're proportional to each other. And that's what we did. And here are all the points. And you see that they form almost a straight line, except for a few stragglers, pretty linear. And so it does look like these are diffusing, not thermal diffusion, not Brownian motion, but this is active diffusion. It's something that simulates ordinary diffusion by means of some kind of random motion. So there has to be some kind of random motion. So it is diffusive, and we get a diffusion coefficient out of that. That's the slope of this line. About 10 to the minus 3 square centimeters per second. So we know something about how they're spreading out. We don't know exactly the details of it yet. We know something. Yes?
Just a question. So if you use the Safman-Delbruck equation, what diffusion coefficient would you expect? Because you say it's not Brownian motion. So how much is it different, the diffusion coefficient, from the estimate from just the size and knowing the viscosity?
I hadn't thought of using that particular way of doing it. Maybe you can explain to me later how you do it, because I haven't run across that.
Okay.
Yes,
Yeah,
thank you.
like 5 orders.
5 orders?
Somebody else do it. Okay, so it does do a kind of diffusion. But how does it manage that? Where's the randomness come from? Well, this is a picture of a single Chlamydomonas. I'll show you a better one later, but this was one done by Marco Poland and Yudan Tuval in Cambridge. And watch this carefully. The two flagella. It's pinned on a pipette, micro pipette. So it's being held. So there you go. You see that?
Whoa.
It's they're synchronized motion, but every once in a while you get an asynchronous. Did you see that? So from this, we begin to realize that these flagella can be synchronized, but they don't have to be synchronized, and they're not always synchronized. Okay? Okay, so now we don't know how it makes the transition from one to the other. We don't know whether it's actively controlled by some kind of biochemical network, but it certainly can make that transition. Yes.
Does that move to asymmetry periodic in any way? Is it kind of like a tumble that you see in bacteria?
We've looked at the statistics of this, and it's not periodic. There's a mean time between desynchronization. There's a mean time, a few seconds. But there are large stochastic fluctuations. It's not a periodic process.
Might it be coupled with whether they are moving down a gradient of light?
Well, we decided to simplify things by doing this in the dark. Okay? Because that brings in additional variables. I mean, you do have, these things are so complicated, but you really do have to simplify. So we haven't done it in the presence of light yet. That's something for you guys to do. Yeah.
Is there a way for them to turn, to change direction?
Yeah.
When they
Well,
unsynchronize?
if they become asynchronous,
They turn, right?
you might think they would turn,
Yeah,
right?
I do think, but I don't know.
Yes. Yes. So maybe that's the mechanism by which they turn. I mean, they have to change direction in order to diffuse. So maybe this is the core idea here that makes it possible for them to diffuse. The fact that they can desynchronize. Yes.
Has anyone measured the persistence length of their? What length scale are they?
Yes. Yes. I'll show you some statistics. Yes.
When it breaks the synchronization of the two flagella, does it do that typically for like an average number of strokes or for a period of time? Or where the
Well, let me show you. Here's a good place where I should show you a little more statistics. What we did was to, I didn't really show you the definition of these things here, but think of these as two oscillators that can go back and forth. Call the left oscillator x sub L, call this displacement x sub L, and the right one xr, and look at their phase relationships. So you can actually get this out of a video image. You can define these amplitudes for the left and right oscillators, those two flagella. And what you see is that sometimes those two signals are in phase with each other, like they are here. And sometimes they're not in phase with each other, and the two frequencies are different by maybe 15%. So it's not only that they're out of phase, but also that their rates are different. So the way the group decided to characterize this was to look at the phase difference between these two oscillators. Can you imagine that? You have two oscillators, and you can measure the phase of each one, and you look at the phase difference. Okay? So if they're synchronized, the phase difference stays a constant, 0 or 2 pi or 4 pi or 6 pi. But on the other hand, if they're asynchronous, then it's not a constant. So this is what happens to the phase difference as a function of time over a minute or so. Okay, so where this line is horizontal, is that horizontal? Yeah. Where that line is horizontal, they're synchronized. And then there's an asynchronous period, like this one, so let's call that drifting. And then it will lock again and become synchronous. And so forth. So you get a mixture of synchronous intervals and asynchronous intervals. That seems to be what happens. But so far, we can see that there's this kind of stochastic synchronization, but there's not yet a very close linkage between this and the turning behavior that Daphne asked about, right? So we need to somehow connect the two. So this is, let's see, I forgot to show you the wonderful apparatus built by Knut Drescher. I don't think what do I do with that? All right. Knut Drescher built a wonderful apparatus with multiple cameras, so you could photograph the organisms from different angles and follow them in 3 dimensions. And so this is an example of one of the trajectories of one of these cells going in 3 dimensions. By the way, here's that spiraling motion. Remember that? So you see that it will generally spiral, but then every once in a while it gets to some point where it makes a very sharp turn. Okay? And that is where we think that the motion became asynchronous. However, we have a problem. We think that, but it's very hard to prove, because when you're tracking these motions over long distances, you can't actually see the flagella. And when you can see the flagella, you can't see the tracks. So you have a problem of how you connect two phenomena that you can't see at the same time. Well, by the way, this is the rate of change of the angle in one of these sharp turns. You get this big peak in the angular velocity, the rate of change of angle. So that's one of these events that we're interested in. So how do you connect these two phenomena? The basic answer is that we came to, or you might find another way. And actually lately we have begun to be able to follow the details of the stroke while we're swimming, instead of having an organism pinned on a pipette. But at the time we did this, we didn't have that capability. But the key idea in connecting these different phenomena was to use statistical ideas. So, for example, we could look at the probability distribution of the time between turns, I'm sorry, the duration of the term. This is the duration of the term, 1 to 4 seconds. And that was measured by looking at the trajectories, the red, the blue. And the red was the time that this phase change took, as measured on this graph, the time of the rising away from the flat steps. Okay, so those are the two phenomena we want to rate, we want to connect, and they have the same probability distribution. Similar, almost the same probability distribution. So that was a piece of evidence in our puzzle. The notion that the probability distribution of the times for these two different phenomena seem to be approximately the same. Okay? So now, depending on whether you're a critical referee or not, you might say, well, that's just circumstantial evidence, or you might say it's very unlikely that that would be true in any other circumstance, depending on how, depending on your how stickily you want to be. But anyway, those are the kinds of measurements that we use to connect these phenomena. Eventually, we had a number of different lines of evidence suggesting that desynchronization is the cause of diffusion. By the way, I want to, in line with Pecco's example, just remind you about dimensional analysis. You know that a diffusion coefficient is always the square of a velocity times the time, characteristic time. So for a random walk that reorients every time tau and travels at speed u in between the reorientations, u squared tau gives you the right dimensions for diffusion coefficient. So we expect it to be of order u squared tau. So we could estimate it just from what we know about the various quantities. We measured the time between orientation events, reorientation events, about 10 seconds. And the typical swimming speed is 100 microns per second. So you can tell that the diffusion coefficient has to be about 10 to the minus 3. The denominator is sort of a small correction factor that corrects for the fact that when it changes direction, it doesn't necessarily go to a new direction that's completely decorrelated with the first one. There can be some residual correlation. So theta is the mean angle change in a reorientation event. But it's not that important. The main thing is square of the speed times the time is an estimate of diffusion constant, where the time is the time between reorientation events. Okay, well that's the first story. I'm telling you some short stories in investigations of this remarkable organism. There are a lot of things we still don't know. We don't know how the cell controls or regulates this behavior of synchronization, And how, so one of you already asked about how this might be connected to phototaxis. These are things we really don't know about. We know something about the macroscopic behavior, but we don't know how it actually works on a microscopic level. One of the striking things about this behavior is that something like it had been previously seen in bacteria. It's called run and tumble motion. How many of you have heard of that? Run and tumble motion? Very familiar. Where E. coli, for example, have two states. One where the flagella are wrapped around each other and spiral, and the organism goes in a roughly straight direction, and then another time interval where they come unwound, the flagella become unwound, and the organism reorients. That's the tumble phase. And the remarkable thing about this work is that it's similar in a sense, that these eukaryotic cells also have two states. And the transition between these two states is related to synchrony, just as happens with bacteria cells, which are very, very different biologically.
Is the tumble part of the bacteria swimming, is it due to thermal fluctuations? So
No.
they stop, it's not?
It's not, I don't think it's believed to be, but I don't know is the actual biochemistry of that is. Okay, so that's the first story. The second, here are the remaining stories that I want to tell you about. So, first was diffusion. The second is velocity fields. Third is mixing. And the fourth is Magellan velocities, Magellan conformations, Magellan mechanics. How the flagella bend and the forces that they're able to exert on the fluid. So these are the four stories. I've said what I'm going to say about diffusion, and I'd like to say something about these other three. So I may just do two of them and then maybe come to this one in the beginning of the next lecture. We'll see how the time goes. Okay? Okay, so, all right. So we want to look in some detail at the velocity field. And the first step in that is to look at the organism's motion, so we can begin to see the effects of this beat frequency. Remember, beat frequency is the period of the motion of the swimming stroke. Well, one of the amazing things about these organisms is they don't just move ahead. If you do the breaststroke and swim, you move at almost the same speed the whole time. And even when you're retracting your arms and getting ready for another stroke, you're swimming forward because of inertia. But there's no inertia here. Rather, when you're not pushing forward, you can't move forward. You actually go backwards. But during the period of time where the flagellum is being retracted, it goes backwards. So here we see see that. And of course, you can do this with, this is really not that hard, this measurement. It's just video tracking of the cell body. And you have to find the cell body, find it accurately, and watch its trajectory over time in a sequence of video images. So what happens is it goes like this, and then back a little bit. Okay? And that's what this velocity versus time picture shows. Okay, so you can see it goes up to 500 microns per second in body. Its average speed is about 100 microns per second, but it can go up to 500 microns per second, and then it can go negative 100 microns per second. It's not really sinusoidal, but it is oscillatory. And you can characterize that. One of the first things you would wonder, I'm sure, if you were doing this, is whether it's periodic. Is it strictly periodic? Is there an oscillator that's controlling the periodicity? I'm talking about during the time when the two flagella are synchronized with each other. Well, the best way to see whether it's periodic is to look at the probability distribution of the frequency. And that peaks at about 50 hertz. But you can see there's a range. And that range of frequencies could have two causes. It could be that you have different organisms. You're sampling hundreds of organisms, and each one is a little different. That's a possibility. But it's also possible that if you look at one organism over time, each cycle is a little bit different from the subsequent cycle, right? And we have not actually looked carefully to distinguish those two. That's sort of something we forgot to do. And so I don't really know which of those are more important. I suspect that both are involved, but we haven't actually looked carefully at whether this is variability, cell-to-cell variability, or whether this, the width of that distribution is instead variation over time. So, okay, well, what I said we were going to do is try to get some idea of the velocity field. If we're interested in fluid dynamics, we want to know the velocity field. Why do we need to know this? Well, now I need to explain to you why we actually have to have this in order to measure the velocity field. We couldn't measure the velocity field without it. And I want to point out that there are a number of difficulties in trying to measure velocity field of one of these organisms. For one, if you have a three-dimensional cell, container, the organisms don't stay in the focal point of your microscope. So they will go in and out of focus, and they swim vertically. You're happy to swim vertically as much as horizontally. And so you have a really big imaging problem if you try to do a measurement like that in 3 dimensions. Another problem is that they don't move in straight lines. They don't move in straight lines, they're moving in all different directions, and somehow you have to measure the velocity field in the presence of all kinds of irregular motion. And the third problem is that the normal way of measuring velocity fields in fluid mechanics is to track particles. So you put some particles in, as I showed you before already, put some particles in there. But to get enough particles in there to measure the full velocity field with high resolution is impossible to do, because you have very little time, and you need some thousands of particles to measure a velocity field. So that would be a very high density of tracer particles. So those are some of the problems that you have in measuring velocity field. Yes?
Your experimental geometry, is it a cube or?
Well, we have done some experiments in not a cube, but in a situation where you might be several millimeters deep and 8 millimeters across, a little disc the shape of a dime. But it turns out to be a bad strategy if you're trying to measure a velocity field. And it's better to try to do it in 2 dimensions. So what I'm leading up to here is to explain why it's advantageous to work in 2 dimensions. So here's the trick that makes it possible to measure the velocity fields really accurately. That is to use a soap film. So you take, this is a real-time picture of these guys. There's a couple that are stuck together. This is a picture of Chlamydomonas cells moving on a soap film. Now, so you make a wire square. Of course, you need some apparatus to do this, but conceptually, the idea is very simple. We didn't invent the idea of making soap films for studying microorganisms. Wu and Duchambeau did that more than 10 years ago. But they were studying bacteria and were generalizing it to eukaryotic cells. But there is a problem here too. Each solution introduces a new problem. The problem here is that these organisms do not like soap. They do not like surfactants because they have a tendency to dissolve the cell membrane. Okay? So you have to find a compromise. You have to find low, sufficiently high concentration that will give you a stable film for a reasonable period of time, like 10 minutes. But not such a high concentration that you damage the cells in that time. So it's a little delicate. But it can be made to work. So that's the first step. Now here's a little bit of the details of how this was done. And this was Jeff Guasto, who's now at MIT, colleague at Pecos. It's still it's time for it to go. So we're talking about a very small quantity of fluid, 2 microliters. We have 1 micron spheres used as tracers. And we're taking images at about 500 frames per second. So we can really resolve the motion detail. Let's see, do you guys get to stretch before I show you the results? Okay, everybody want to stand up for a moment?
We're in suspense.
What?
We're in suspense.
In suspense, all right, here. So let's see what we get. So here's some swiveling trajectories in the film. By the way, it might be a little different in the film than in 3 dimensions because they can't spiral. If the film is maybe 10 or 15, say 15 microns deep, flagella are about 10 microns long. That's long enough that they can't turn over. So their response to light might be different in this environment. Something to keep in mind. Anyway, you see these trajectories, and one of the things that's most evident about that is that doesn't look like a random walk, right? It looks like what we call ballistic motion. It's smooth motion. But over a long period of time, it becomes more like a linear. Over a short period of time, it's okay, now here comes the velocity field. Now, we're going to use these tracer particles to measure the induced velocity field. Well, first of all, you can see that there is an induced velocity in the fluid, right? You can see that because the tracers are moving, as indicated by the red tracks that we found for the tracer. So every time a cell passes near a tracer, it gets a little kick. Okay? But how do you use that information to reconstruct the velocity field of the particles? That's the thing that's difficult. So here's the, I don't know if you can figure it out, but here's the trick that Jeff devised. His idea was that since you know what the body is doing, and it's going like this, you have a built-in clock. And you can look at the organism every time, let's say every time it gets to its maximum displacement. Every time it has a particular phase, think of it as an oscillator. Every time it has a particular phase, you look at the particles. You wait till the next time. You look at where the particles are. So on each snapshot, you really don't have enough particles. You have only, maybe you have 10 or 20 particles near the cell. Not enough to get a velocity field. But if you do this for 1,000 times, 1,000 periods, you'll multiply the number of sampling particles by 1,000. So you might have 10,000 particles altogether by using that trick and taking advantage of the periodicity of the beam. In that way, you can build up the spatial resolution that you need to measure the velocity field. Oh, I didn't know this was on here. I guess, you see that? There's a problem that you have to overcome, which is that they're also turning. But you have to, there we go, there's the second one, there's the third one, there's some more particles. And so now you're beginning to get enough particles to measure the velocity at that particular phase. Which you also have to do with every other phase as well. Yes?
You were saying all the time that most of them are periodic.
Oh, if it's not periodic.
You were saying.
Yes, it's not quite periodic, but it's close enough to periodic to make this work. So for one organism, yes. Okay. The fact that this works for one organism suggests that it's the cause of the broad frequency spectrum is variation from one organism to another. Whereas if you stick with one organism, it's more nearly periodic. Yes?
There is no influence of the other swimming organism?
Well, they have to be far enough apart that there isn't. If the density is too high, this also wouldn't work. Other questions? Yes?
So I guess this is limited to the flow field near the particle, right?
Yes.
You can go there.
Yes.
Which is fine.
Yes. Basically, the flow field falls off as about 1 over r. And so you can go a certain distance out, you lose resolution, and you really can't realistically go more than 30 microns from so there is a limit on the far out. Well, let's see the result. We don't need all this stuff. This is showing the sampling once per cycle. And then, so you measure the velocities at each phase, then you can average them I'll show you the result averaged over time, and then not averaged over time. So this is what it looks like averaged over time. This is the time average velocity field near a swimmer. Let's take a look at it. Well, so you see velocity vectors, which we have sketched out here. And you see these vortices, two vortices in the time average velocity field, which are very strong. That's they're being forced. You normally wouldn't expect vortices in a low Reynolds number flow, but if it's forced by the motion of a surface, then you can have something. So, but we didn't expect that. We were surprised at these very strong vortices. I should say that this is the body of the organism right there. And we've drawn it in a way that it's instantaneously pointing to the right. It's instantaneously moving to the right. We're not looking at this in the breast frame of the organism. This is the lab frame. But we fix the orientation. We turn all our pictures around so that they all point in the same direction. What about this spot right there, that green spot?
Stagnation point.
There's a stagnation point right there, where has the topology of the saddle point. The green stagnation is visible in the plot. So that's the time-independent velocity field with a resolution of about 3 microns. And I guess we're going out about 40 or 50.
Can I ask a question? Maybe you answered it already, but so is this the field, the instantaneous field at this specific point in the cycle? Or is it an average flow field over a cycle?
So we took the data at each phase and we averaged them all together to get the.
So it's an average over a specific point in the cycle, right?
It's the average over all times. So it will become clear when I show you the next picture. Okay. Let me show you. Oh, let me show you the time components.
Oh yeah, that's what I meant. Okay.
There's the time components. So, and let's see, look at this picture. It helps to watch this little organism, show you where in the cycle we are. And this is showing the body, the velocity of the body. Okay, forward, backwards, and so forth. And this is where in the cycle we are. And then we have to look at the shape of the velocity field to see. Okay, so that's the forward stroke. And now, right there, it's the flagella retracting. One second, retracting. And you see that these vortices are quite different. It's more nearly symmetric. There are 4 vortices at that particular time. Yes?
Did you also see the flow of particles when you were holding the algae with the pipette tip?
No, there was no velocity there. Just the phase was measured by light intensity variations from the flagella. Yes.
Comparing these flow peaks or the order of magnitude of these flow peaks with just random Brownian motion, do we now estimate how far away these pitches feel each other?
Yeah, how far away they can feel each other.
To feel their respective velocity.
Yes. In fact, well, of course, it's it doesn't have a definite answer because there's a weak effect that goes on quite a long distance. In fact, let me show you that. Let me show you the fall off of the velocity field as a function of the distance from the center of the body. So I'd like you to look at the green. Look at axis. And this is going off the back of the organism. So it's swinging like this and then going in the backward direction. It shows you the fall off of the intensity. This is in the average velocity. Look at that. The solid line is 1 over r. So it's about 1 over r. Now, be with me a second. It's about 1 over r. If you do this in 3 dimensions, you get a faster fall off. Velocity fields fall off as 1 over r squared. But it's actually 1 over r. So you ask, how close do they have to be to feel each other? Well, it gets weaker and weaker as 1 over r.
There must be one point when just random fluctuations are
you want to know how far away until the thermal fluctuations become as large as these. That's your question. That's a very well-defined question. And that answer, of course, is also different in 2 dimensions than in 3. Right? I forget what the number is here. In 2 dimensions, it was about 25 microns. In 3 dimensions, it was about 25 microns. Here, it would be a little far. But I don't know the number. Now, what about these other 2 lines, like this one? Why is dip here in the velocity? Well, that's where you go through the stagnation point. Right? If you go this way, you're going to get to something close to 0. Right? On the other hand, if you go this way, there's also a stagnation point right here. The centers of those 2 points. So those dips are understandable also. But the most important thing to remember is that they fall off. It falls off as 1 over r. That's pretty amazing. It's very long range. Relative, and that's what we mean by a long range force. A force that falls off is 1 over r. It's generally considered.
The length scale Sebastian asked you for should also give you some concentration threshold for the possible collective behavior between the swimmers. Because if they don't feel their respective flow, they cannot.
Yes, so you could estimate.
Is it related, or did you measure?
I think it's a good point. You could estimate a minimum concentration. We have looked at concentrations up to 7% solid, which I think is high enough that if we had collective motion possible, we might have seen it. But we're still working on it. That's something that's on our list to do it better. We really don't know. And there are some theorists who say that polar organisms like this won't produce collective motion, whereas the pushers like the E. coli will. I don't myself understand the argument very well, but there are claims that it won't happen, but there's no really good experiment yet. Yes.
So as you point out, the flow field is significantly different in 2 dimensions. So do you have any reason to believe that the gates are going to be sort of modified significantly by the difference in the flow structure?
2D versus 3D. Well, qualitatively, they seem to be fairly similar, but quantitatively, you're probably right. On the other hand, we gain tremendous advantages in instrumentation by doing it in 2 dimensions, and so you have to, you get some, eventually in the long term, perhaps both will make a contribution. Let's see, I want to bring this part of the talk to a conclusion because we're very close to 9:30. This is the sort of 6 snapshots at different points in the cycle, but we've sort of talked about this. But the most important, I think the most interesting thing of all we've not talked about, and that is how much energy is being dissipated in the fluid by this induced velocity field. Well, we can calculate that because we have the whole velocity field. In fact, that's one of the most useful things about having the velocity field. You can get the energetics. How do you do that? Well, here's how you do it. You measure the velocity gradient tensor, you take its square, multiply by the viscosity, and integrate over the space surrounding the organism and over the thickness of the layer. So you can actually calculate the power. This is standard fluid dynamics. Right? Velocity gradients tell you viscous dissipation, basically. So you can measure the viscous dissipation. Just if you know the velocity field, you can get the viscous dissipation. Gamma is the velocity gradient. So that's what you get. 15 femtowatts peak. You know what a femtowatt is? 10 to the minus 15 watts, okay? For one cell, 10 to the minus 15 watts. So as a function of time during the period, it goes up to a maximum of 15 femtowatts. Now if you pick a different organism, it might be 20. But the order 15. And then it falls off, and then you get another peak. What's this other peak? That's the backwards motion, right? Smaller peak. But you're still putting energy into the fluid. That's wasted energy. It's not doing anything. It's taking you backwards. So that's the pattern. 15 femtowatts peak. And here's something that I find really charming. Look, the average, if you average over time, over a cycle, 4 femtowatts is the power. The mechanical power that's being dissipated in the fluid. But if you look at the mean flow, you look at this velocity field, this one, the time, the mean flow, and ask how much power does that dissipate in the fluid, it turns out to be 1 femtowatt.
So Jerry, how many can you put in your liquid? How many guys? So that they're still happy.
Well, think of this as one isolated cell.
Yeah, I'm just trying to scale it up. So if you, how many can you put in? A femtowatt is pretty tiny, but if you put in 10 to the 15th,
okay. Let's say, well, you can certainly get 1% solid. So I don't know if you could do that. I'm reluctant to go on by now. So look, the main point I was making is that if you could use, if it didn't oscillate, if it didn't have to go backwards, it would only need 1 femtowatt. But in fact, it needs 4. That's just because it has to go backwards. Why does it have to go backwards? That's because of the scalar theory, right? It has to have a different backstroke and forward stroke. And it's overcoming that requirement that is the waste that Pecco talked about. She said it's just inherently inefficient to swing the load elsewhere. And this is a quantitative measurement. Okay, so maybe, yes, I'm just about done. Look at this. Where's this coming from? Where's this energy coming from? It's coming from the hydrolysis of ATP, right, in the molecular motors. You can estimate that the power production that we've got here requires metabolizing about 1 ATP molecule for every 15 nanometers of flagellar length per cycle. But we can say something about what's going on inside the flagellum. Not very much, but something, neglecting any direct heat production. Okay, so that's where I'm going to stop for today. This is what we know based on the velocity field. And I think I will spend about an hour of the next talk on these two stories. One about the mixing that's produced, and one about the actual forces generated by the flagella. And I anticipate having another half hour that I can use for something completely different. And I'm still debating about what that would be, but it might be something about No, I won't tell you. It won't be research topic, it will be some other aspect of biofluid. Well, thank you very much. Okay, let's have some coffee.